binary fission in amoeba and budding in hydra. The aim of the experiment is to study binary fission in amoeba and budding in hydra with the help of prepared slides. The materials and apparatus required to perform this experiment are prepared slide of binary fission in amoeba, prepared slide of budding in hydra, and a compound microscope. Before we start with the experiment, come, let us revise some theory related to it. Reproduction is one of the basic characteristics of a living organism. There are two types of reproduction, sexual and asexual. Sexual reproduction involves both male and female gametes, while in asexual reproduction, no gametes are involved. Asexual reproduction in living organisms are of various types, such as binary fission, multiple fission, budding, fragmentation, etc. Come, first let us study about binary fission in amoeba. Observe the prepared slide of amoeba showing binary fission first under low magnification and then under high magnification of a compound microscope, and note down the observations. When we observe the slide of amoeba showing binary fission under the compound microscope, we see that binary fission in amoeba starts with the division of nucleus. The cytoplasm shows the formation of a transverse furrow after the division of nucleus. This furrow deepens and divides the parent amoeba into two daughter cells. Now, we will study about the process of budding in hydra. Focus the slide of budding in hydra first under low magnification and then under high magnification of a compound microscope and note down the observations. When we observe the slide of budding in hydra under the compound microscope, we see that bulb-like projection called bud arise from the lateral side of the body wall. This bud then gradually increases in size and volume by division of cells and forms a young hydra. This young hydra, after its complete development, gets detached from the parent body and starts an independent life. Thus, based on these observations, we can infer that both prepared slide of amoeba and hydra show asexual mode of reproduction in which only one individual is involved in the production of new individuals. During the process of binary fission, the parent's identity is lost, while during the process of budding, the parent's identity is maintained. Come, let us now discuss precautions to be taken while performing this experiment. The only precaution to be taken while performing this experiment is to focus the given permanent slides properly under the compound microscope so that the observations are clear and appropriate.